All right, welcome back everyone. So we're going to pick up here and we're going to begin talking about lipids for the next few moments. So lipids, you may think of them as fats, right? These are going to serve various functions, as we'll see in just a moment. Functions with regards to the structure of our cell's plasma membrane and creating hormones and synthesizing various products. So there's different types of lipids that we'll talk about. We'll talk about triglycerides. We'll talk about the kind of some of the parts that make up triglycerides called fatty acids. We'll talk about phospholipids. We'll talk about cholesterol, things like that, right? So with fats, this first type that we'll look at is called a triglyceride. So when you think of fat, you're probably thinking of triglycerides, right? So triglycerides have a structure of a what's called a glycerol backbone. And then extending off of that glycerol backbone are three fatty acids, right? So fatty acids are these long chains of carbons with hydrogens attached to them, and they exist in a couple of different forms, right? So one is, excuse me, is the saturated fatty acid, right? So this term saturated, and likewise the term unsaturated, it's referring to this backbone here of carbons and whether or not there are hydrogens chemically bonded to those carbons, right? So if you have a saturated fatty acid, simply this means the carbons are saturated with hydrogens, right? So that's where that term comes from. Saturated, the carbons are saturated with hydrogen. So that meaning at each point there is a hydrogen attached. Okay. For unsaturated, you can probably imagine that this means that some of the carbons do not have a hydrogen, right? So meaning that they are not fully saturated, and so you call it unsaturated. So this one on the right that you see here, this is an example of an unsaturated fat. Here at this point, this double bond here, this covalent bond, there's not a hydrogen. So it produces this kind of bend in the fatty acid chain, right? In this unsaturated fatty acid chain. Now, you find these different fats in different, or they come from different sources. So you find them in different areas, right? So saturated fats typically come from animal sources, okay, and are typically solid at room temperature. Okay, so this is the fats that you find in cheese, milk, meat, right? All of your animal sources, and then various types of oils like coconut oil, palm oil. Your unsaturated, these are often from non-animal sources. Okay, so other types of oils, nuts, things like that, right? These are unsaturated. Okay, so meaning again, there's at least one point on that fatty acid where there's not a hydrogen. If there's only one point, that's called a monounsaturated, and if there are two or more, then you call that a polyunsaturated fatty acid. Okay, so there's a third type here called trans fatty acids. Okay, trans fatty acids are similar to the saturated fatty acids, but in this case they have been modified through this process called hydrogenation. Right, hydrogenation. So this is changing the position of some of the hydrogen atoms. It essentially kind of switches the orientation of them. So instead of being on the same side, they move to opposite sides. What it does is it protects the fats. It essentially prolongs their shelf life, right? So it's used in corporations and businesses to prolong foods' shelf life. However, it does make them behave more like saturated fat. So they're extremely unhealthy, right? Trans fats, trans fatty acids, right? And so increased amounts of saturated fats, increased amounts of trans fats are associated with higher incidences of cardiovascular disease, heart attack, stroke, things like that, coronary artery disease, right? So a second type here is called phospholipid, right? So we've talked about phospholipids before. You're likely familiar with these. These are made up of a few different things. One is a head, so they have this phosphate head that you see here, and then they have these two fatty acid tails, right? And so these fatty acid tails, these are the same fatty acids that we just talked about, right? So one here is maybe saturated, one is unsaturated, right? Now you may also recall that we described some of the properties of the head and tails, being that the head is called hydrophilic and the tail is hydrophobic, right? So remember, hydrophilic is meaning water loving. It likes to be in contact with water. Hydrophobic means the opposite. It fears water. It does not want to be in contact with water, right? So the heads are polar. These tails are non-polar, okay? So this term amphipathic refers to a molecule that has both a polar and a non-polar region, right? So meaning, in this case, the head is polar, the tails are non-polar, it has both, therefore this is an amphipathic molecule, right? And phospholipids in our diet food are used as emulsifiers, right? So allowing water and oil to mix. So think of salad dressing, think of like in things in mayonnaise, candy bars, things like this. Okay, and then again, as we mentioned, main constituent of our plasma membrane. It's not the only thing that makes up the plasma membrane, but it's a big portion of our plasma membrane. Right. Now the last type of lipid here that we'll look at is called the sterol. Right, sterol. So these are a class of steroids that they're in kind of this ringed structure, and a good example is cholesterol. Right, so we've all heard of cholesterol. You've likely heard of this. This is a product, a nutrient that comes from animal cell membranes and not plant cells. So you only have it in animal cells. We are animals as humans, so therefore we have cholesterol in our cell's plasma membrane. And in our membrane, it helps to kind of stabilize the membrane and keep it steady and stabilized under temperature changes and things like that. However, we do know, and we'll talk about this later, that you can have too much cholesterol. So cholesterol levels in the blood can become too high, and this is going to affect our health, and again, put you at an increased risk for cardiovascular diseases, things like that. This is the approximate blood cholesterol level. That is a recommended number, so under 200 milligrams per deciliter. Now, we also get cholesterol in our body naturally, right? So our liver can synthesize cholesterol through something called beta oxidation that uses this enzyme. And this enzyme, as we'll see in a little bit, is the target of a drug or a class of drugs called statins. Right? Statins are drugs that help to lower blood cholesterol. Okay, help lower blood cholesterol. Another thing associated with cholesterol is kind of what we talked about previously whenever we did carbohydrates, is that fiber can help to lower cholesterol. Right? So when the fiber is there, the bile that contains the cholesterol, remember bile is produced in the liver, is not as able to be reabsorbed, and so that bile, along with the cholesterol in it, is passed in our waste, in our stool, and so that cholesterol is not getting reabsorbed. So there's evidence that fiber helps to lower cholesterol levels. Okay. So these are the general functions. Again, membrane, parts of our cell plasma membrane, right? So the phospholipids, myelin sheath, right? So if you recall, when you've talked about neurons and action potentials and nerve impulse conduction, the myelin is this kind of fatty covering that wraps around the axons of our neurons, insulates them, and allows the electrical signal, the action potential, to move more quickly, right? And so the cells that do that are the myelinating cells. These are like the Schwann cells and the oligodendrocytes. They wrap their membrane around the axon, and their membrane is lipids, right? It's phospholipids. So we also have this for the absorption of fat-soluble vitamins. E, D, A, and K are B vitamins, and our C vitamins are water-soluble. Necessary for producing vitamin D. Lipids are necessary for producing various types of hormones. So think lipid-soluble hormones like testosterone, things like that. Protective function around organs. So around our kidney, we have lots of fat called perirenal fat. Lots of fat behind our eye, things like that. And then energy as well. So 9 kilocalories per gram, whereas our other macromolecules like carbs and proteins are only providing 4 kilocalories per gram. So we consume our lipids, and now we need to digest them, so we're going to use enzymes just like we used for carbs, and so we're using an enzyme called lipase, right? Lipase. So that LIP, that's referring to lipids. So one of it is lingual lipase. This is coming from the saliva in the mouth, and so that lingual refers to tongue. And then, like we had pancreatic amylase, we also have pancreatic lipase, right? Now, if you're looking at this image on the right side of the screen, which part, well, first off, what is this tissue, right? What is this tissue we're looking at? But then what part of the tissue is the pancreatic lipase coming from, right? So you can probably guess, since we're talking about pancreatic lipase, this is the pancreas. These light colored regions that you can see every so often, those are the pancreatic islets, right, or the islets of Langerhans. That's the endocrine part of the pancreas, right? So producing insulin, glucagon. Everything else, all of these, all of this purple tissue, this darker purple tissue, that's part of the exocrine pancreas, right? So the exocrine pancreas, think more along the lines of digestion, producing these enzymes, pancreatic amylase, pancreatic lipase, and then the pancreatic juices that enter the small intestine. Okay, so we produce this pancreatic lipase and we continue breaking down and digesting lipids, and then we're also using the bile that's coming from our liver and our gallbladder, right? And so this bile is necessary to emulsify fat, so separating the droplets of lipids into smaller portions. This is called emulsification, right? And this is done with the bile through the contents of the bile called bile salts that are amphipathic, so like these phospholipids here. And what they do is they organize themselves into this kind of complex three-dimensional structure called a micelle, right? And this is going to transport ultimately allow the transportation of the lipids into our cells. And then, as we'll see, we're then going to move that out of the cell and into a lymphatic capillary. Okay, so these tails line up around our lipids, creating this micelle structure, right? So here you can see, hopefully you can see on this right here. So this is this space here. This is the lumen of our intestines. And so what we have is we have our fat, we have the bile. The bile kind of separates this large fat droplet into smaller droplets that are then coated with those bile salts, right? So emulsified by the bile. And these are the micelles, and then these are then brought into the intestinal cell, right? So this orange cell, this is a cell lining our intestines, right? And so what happens here is this micelle is brought in, and it's wrapped in cholesterol, and it's wrapped in other lipids, and it's wrapped in proteins. And as a result, you form something called a chylomicron, right? A chylomicron inside this Golgi apparatus, right? One of our intracellular organelles, right? These chylomicrons then move out of the intestinal cell. They're exocytosed, right? So they're being released, and then they are brought into this green structure, which is a lacteal. If you recall, a lacteal is a lymphatic capillary, which is part of our lymphatic system and transports lymph. Okay, so in this case, the chylomicron, which again is the cholesterol and lipids wrapped around with protein, wrapping around our triglycerides, our fats, are brought into the lymphatic system, and then they circulate in the lymph until the lymph kind of dumps the lymphatic capillary dumps the fluid back into the bloodstream, right? So where the thoracic duct and the right lymphatic duct join back in the bloodstream. Okay, so this here is showing a little bit of the kind of the pathway of lipids in our body after it's being absorbed, right? So again, as we mentioned just previously, you're first absorbing the fat, it forms the micelles, those get converted into these chylomicrons that then get brought into the lymphatic capillary. So that's what we see here with number one. From here, we're going to have lipases break down these chylomicrons into their constituent parts, so the fatty acids we mentioned earlier and the glycerol backbone. So again, think of the components of the triglyceride that go into the cell. These chylomicrons go to the liver, they get broken down and degraded. And then we have these what are called lipoproteins. Okay, lipoproteins. So these are transporting lipids in the blood because, of course, our lipids are not water soluble, right? So our blood is, the plasma is mostly water. These are lipids. They don't mix, right? So these lipoproteins are aiding in this transport of lipids in the blood. Okay, so we've got a few different types. One is called a VLDL. Okay, this stands for very low density lipoprotein. VLDL, very low density lipoprotein. to give you a easy way to remember this, the density, and we'll talk about two others called low density lipoprotein and high density lipoprotein. The more lipids that it has, the lower the density. So very low density lipoprotein has lots of lipids. Low density lipoprotein has still a lot of lipids but less than the VLDL. And then high density lipoproteins, HDL, contain the least amount of lipids. Okay, so we're going to transport fats and triglycerides to our body cells. So we'll do this with VLDLs and LDLs. Okay, so when you think of these two, VLDL and LDL, think going to the tissue. So going from the liver to our body's cells. Right? That's what LDL and VLDL are doing. They're taking the cholesterol, the fats, the lipids to the body cells. Right? You then have one called an HDL, high density lipoprotein, less lipid, more protein, and it's doing the opposite of LDL and VLDL, meaning it's transporting the fats, lipids, triglycerides, and whatnot from the tissue back to the liver, right? So we're moving it in the opposite direction, okay? So you may hear of things like good cholesterol and bad cholesterol. That's kind of what we're talking about here, with LDL being the quote-unquote bad and the HDL being the quote-unquote good. Okay. This graph here is showing basically the same thing, but in a little bit more simplistic way. So here's HDL, again, going from our tissues back to the liver. So this is a hepatocyte, a liver cell. And then we have VLDL and LDL leaving the hepatocyte, leaving the liver, and going to the tissue. So you can see here in blue, we're going from the liver to peripheral tissues, whereas here in green with HDL, we're going from the peripheral tissues ultimately back to the liver. So here it's picking up cholesterol, incorporated into the HDL, and then it makes its way back to the liver. Okay. So there are considerations for having, a certain cholesterol level. So typically, if something is above 200 milligrams, if your blood cholesterol level is considered above 200 milligrams per deciliter, that's considered high. And having elevated LDL and or decreased HDL puts you at an increased risk for some type of cardiovascular event, right? So think stroke, heart attack, coronary artery disease, something like that. So there are medications that can be given to lower cholesterol, and these are called statin drugs. So they all end in statin. And so what these do is these are inhibiting an enzyme called HMG-CoA, which is essentially synthesizing cholesterol in our liver. And so if you can provide a medication or a drug that blocks these this enzyme, then you're going to decrease and lower cholesterol synthesis, right? Thus helping out with overall cholesterol levels, right? And so again, these drugs, they end in statin, so atorvastatin, fluvastatin, lovastatin, there's a bunch of different ones, but they block that HMG-CoA reductase enzyme that's needed for cholesterol synthesis in the liver. So here you can see kind of a panel of different optimal, near optimal, borderline high, and high risk levels for cholesterol, LDL, HDL, and triglycerides, right? So notice for cholesterol, LDL, and triglycerides, you want it to be under a certain number, right? So cholesterol less than 200 milligrams per deciliter, LDL under 100, triglycerides under 150. For HDL, you want it to be above a certain number, right? Typically. Above 60 milligrams per deciliter, indicating, remember, it's moving the cholesterol back toward the liver. Okay, so anytime you have high LDLs, high cholesterol, high triglycerides, and low HDL levels, then that is an indicator that you're at a higher risk for some type of cardiovascular event, some type of heart attack, stroke, coronary artery disease, something along these lines, right? So if you have these elevated, cholesterol, LDL, and decreased HDL, you can end up with something like this. So this is a blockage in a blood vessel. So this is the LAD. This is the left anterior descending artery. It runs down the anterior surface of the heart in that interventricular sulcus. Recall it's a branch of the left coronary artery. And so you can see here on the left that there's a significant blockage of the of the artery. So you can see that constriction there. This is plaque building up on the inside of the vessel, narrowing the lumen, right? So what can be done is you can place a stent. Okay, so a stent is this kind of metal mesh-like structure. It's fed through an artery, usually, maybe starting in the groin, and it goes backwards. The stent is traveling backwards under the guidance of a surgeon. It's moving backwards toward the heart, into the aorta, into the coronary artery, and then into whatever branch is blocked. So in this case, it's the LAD. The LAD, it's also sometimes called the anterior interventricular artery. Okay, kind of in general terms, and when it's blocked, this is called the widowmaker, right? Because blocking this blood vessels can significantly impact and influence blood flow to the heart, causing a heart attack, right? And so this stent, again, it's fed backwards in an. Remember, it's got to be in an artery. If we go backwards in a vein, or rather forwards in a vein to the heart, we're going to end up in the right atrium, right? And that's not where we want to end up here with an artery. We want to end up in the aorta, going back into one of those coronary arteries. And in this case, we'll go to the left coronary artery into this. And then the stent is expanded. There's a little balloon that expands and opens the stent, and it pushes the plaque against the wall, and it opens and restores the patency, the blood flow in that blood vessel, restoring blood flow to the more distal areas, right? So this is sometimes done. You could also have a bypass graft. So you could have a CABG, a coronary artery bypass graft, in which you reroute blood flow around the blockage. Typically, after something like this, patients are going to go on blood thinners, right? So going on blood thinners now increases your risk for bleeding. And so that's now a whole nother set of issues that you have to worry about. If you had a stent placed and you go on a blood thinner, but then you find out you need surgery, well, you can't have surgery if you're on a blood thinner because you're at increased risk for hemorrhage and bleeding. So there's other considerations that need to be made after a stent placement and going on blood thinners, right? So we're going to stop here, and we'll pick up in our next part, and we will begin talking about the last major class of macromolecule, which are proteins.